Right. Well, as I say, thanks very much for the invitation to talk. What I'm going to do today is tell you a little bit about the way in which plants develop. So in my lab at Durham University, we're interested in mechanisms of plant development, molecular mechanisms. And what I'm going to do is break down the talk as is on the current screen, which is to introduce multicellularity, as plants are multicellular, most of them. I'm going to talk about how, the focus of the talk really is about how plants and animals develop shape in quite different kinds of ways. And one of the features of plant growth and development is that it's a modular system. And I'm going to explain why I think it's modular, what I mean by modularity, in the course of the talk. I'm going to talk about how the environment is particularly important in controlling different aspects of plant development, more so than is the case in animals. I'm going to then move on to talk specifically about roots, something that a lot of people don't talk that much about because they're hidden under the ground mostly. And so it could be considered the sort of hidden half of plants and plant development. Introduce very briefly, some of our work on that. I'm not going to really show any data in this talk at all, more conceptual. But I will introduce some of the experimental approaches that we use, and that includes use of mathematical modeling. This is a more recent kind of approach to understand the gene and signaling networks that control the activity of particular cells that are involved in cell division in roots, and then finish off with some applications of these ideas and where things are moving in the future. So I'm going to start off by introducing multicellularity. And one of the things about multicellularity is that it gives you a certain level of complexity in your final form, whether you're a plant or an animal. And the form of plants has certain features that are actually quite common between plants and animals, such as, for example, the development of symmetry. So bilateral symmetry, where you've got two halves of, in this case, Grumpy Cat's face, which are effectively mirror images of each other. And we see that in plants like orchids, in humans. But then there's also other forms of symmetry like radial symmetry. So this flower here has symmetry across more than one axis. So this thing here, Saccharitis, is actually the most ancient human ancestor. And it was discovered in fossils that are about 540 million years old. And what it shows is bilateral symmetry. So if you draw a line of symmetry from top to bottom, you can see that the two sides are symmetrical. And that's something that really is therefore an ancient phenomenon, the development of that type of symmetry, dating back even 500 million years or so. Now, one of the kind of interesting things that we see in some plant species is that multicellularity is not in itself an absolute requirement for morphological complexity. So these algae here, acetabular area on the left, and these algae on the right, these so-called siphonous algae, although they look quite complex, they're not in fact multicellular. These are single cells. So even though they've got sort of really intricate structures, it's actually a single-celled organism. And so that does raise the question of why you need multicellularity in order to develop complexity. And essentially, I think the reason is that you can develop these massive structures like this giant redwood, which is in fact this particular one, General Sherman it's called in California, is the largest living organism in the world. So you develop massive size through multicellularity. You can't have a single cell that is that big, 200 feet tall. And of course, humans have incredible sophistication in the way they develop different tissues through multicellularity. So one of the big challenges in biology is understanding how you get from a single cell, whether it's a plant egg cell here or a mammalian egg cell, how that develops into these complex multicellular structures. And the way in which plants and animals do that is quite different. And that's kind of the focus of this talk. I want to consider the differences in form between plants and animals and how they're generated. And one of the features, of course, of animals and the way they grow is that they do this in a kind of determinate way, what's described as determinate. And what that means is that the final structures in place, the limbs and organs, and the shape of the animal is generally pretty predictable. You know that this frog's going to have, 2 arms and 2 legs and however many toes it's got, etc. Whereas that is not the case with plants. So although we might understand the overall structure of a plant as being characteristic of a particular species, we're not going to be able to determine early on how many leaves or branches it's got. That's much more unpredictable. And similarly, the final form of the plant is quite plastic, quite variable between individuals depending on environmental conditions. So, for example, this is a sort of a diagram of the root system of a tea bush. In fact, one of which the top here is under drought conditions, and this is irrigated. And you can see the architecture of that root system is quite different, and that's been determined by environmental factors, which I'm going to talk a lot more about later. But not all aspects of plant development is organized in that way. Yes, leaves and branches are quite unpredictable, but floral organs are not. So these are highly stereotyped for a particular species, highly predictable. And in fact, so much so that the taxonomy of plant species is governed primarily by the organization of the floral organs. It's so predictable. And that's why you have a flora of the British Isles or the flora of Europe or the flora of North America. That flora represents the fixed nature, pretty much, of floral organization in a particular species. And there's a good reason for that, which is that you need your floral organs for reproduction. So if you develop floral organs that are, too long or too short or not in the right position, they may not either be pollinated by insects, which have co-evolved to allow pollination in the expectation that the anthers that carry the pollen are going to be in the right position, or self-pollination or wind pollination. You need those structures exposed in order to attract pollen if you're a stigma surface. So there are some aspects of plant development that are quite variable, and others that are not so variable, like flower development. Now, plants and animals have quite different strategies for developing this multicellularity. In animals, of course, a lot of shape is generated by cell migration. And we see this in an absolutely beautiful way in the way in which tissues, layers of cells move around in the developing embryo. And that creates three-dimensional shape through this cell migration. But plant cells don't do that. They don't migrate. And shape is therefore generated not by the migration of cells, but by regulated control of both expansion of cells, so basically filling up with water, and that water having a turgor pressure that pushes against the cell wall that bounds the cell, and then the plane of the new cell wall that's formed, the division of that expanded cell. And that is the mechanism by which plants generate shape, not by cell migration, but by its regulated division and expansion. So when we see a seedling germinating and growing out through the soil to get to the light, the growth of this hypocotyl, this region here, involves no cell division at all. Actually, it's by cell expansion from a prepared during embryogenesis pattern of cells that then just fill with water and expand quickly to get to the light as fast as possible. So the time it takes for the cells to divide is not a kind of a barrier to getting to the light as fast as possible. Now, in animals, cell division occurs in a different way to the way it occurs in plants, and it occurs in a so-called diffuse way. So cells divide throughout the three-dimensional structure that is a developing embryo and ultimately a little mouse. And cell expansion doesn't really contribute to growth in the same way that it does in plants. The cells don't fill with water and generate shape in that kind of way. And in plants, the sort of characteristic defining feature, and which is really the focus of this talk, is the activity of small groups of cells at which cell division for growth occurs, and these are known as the meristems, which of course animals don't have. So this is my diagrammatic representation of where some of the main meristems are in a plant. This is my diagrammatic representation of a simple plant. And you have a group of cells at the tip here, at the shoot tip, which will divide to form leaves and other aerial structures. You have meristems at the tip of the root, at the side of the root. Lateral roots develop here, and I'm going to talk a little bit about that. And then there's a cylinder of cells through the body of the plant called the cambium, which gives rise to radial thickening. But these are the areas, the main areas of cell division. And growth is a sort of a combination of the activity of this very localized cell division followed by expansion of cells away from that site of cell division. And so understanding how these meristems work is a kind of a major sort of biological problem to be solved. Now, this nature of the meristems leads me to this sort of idea of plant growth being a modular process. And what I mean by that is that you can take a meristem, so a millimeter or so of a root tip, for example, you can chop it off and you can place it in a culture medium, which has just got some simple salts and vitamins, and it will develop as a root from that original 1 millimeter or less of tissue. So you get a perfectly formed root from that original structure. And you can do the same kind of thing with a shoot. You can take the apical millimeter of a shoot, and it will then grow to form a perfectly normal looking entire shoot. So this is what I mean by modularity in development. Those meristems have the ability to generate the entire structure adjacent to it, like the rest of the shoot or the rest of the root. And so this modular development is possible because of these meristems, which are peculiar to plants. So they're central to growth strategies. The meristems are a very characteristic kind of tissue. So the cells of these meristems are small, and importantly, they're not differentiated. So they are, as we'll see, I'll show some pictures in a minute, they all look the same. They're small, small vacuoles, undifferentiated. These are the sites of cell division, and they contain within them stem cells. I mean, I don't mean stems as in a plant stem, I mean in the animal stem cell sense. In other words, these are self-regenerating cells that retain this undifferentiated state, this stem-like condition that are able to divide, and one of the daughter cells will then go on to expand and differentiate into a particular cell type. And these stem cells in plants are very long-lived. And a nice example is these bristlecone pines, which are still able to produce cells from their meristems after 5,000 years. These are in the southwest United States. And just so there's a very interesting question there about aging and why these stem cells maintain viability after such a long period of time, but they do. And that's sort of there's an interesting question there about how that's organized. So I want to just say a little bit about the different types of meristem that there are in plants. So we'll start off with the shoot apical meristem here. So this is what a longitudinal section through a shoot apical meristem looks like, and you can see that the cells all look the same sort of shape and size. They're not differentiated, and they divide very slowly, particularly in the center of this meristem. When you move away from the center of the meristem, laterally you move to cells that will start to differentiate, and these are the emerging new leaves that form off the flanks of the shoot apical meristem. And down below the apical meristem are cells that you can see are starting to expand and differentiate, and they will form the vascular tissues, the xylem and the phloem, the water and hormone and sugar conducting tissues that will transport sugars, for example, from the leaves where they're photosynthesizing all the way down to the roots. So you have this group of cells, the meristematic cells, that retain this undifferentiated stem-like condition. And it's very interesting to understand how that stem-like state is retained. And there's quite a lot understood about the molecular basis of that. At the other end of the plant, you've got the root apical meristem. This is a confocal microscope image through a root meristematic region. What you have down here is the root cap a bit further down. So these cells are differentiating, but this group of cells here, you can sort of see these slightly larger cells. Those are at the sort of heart of the meristem. These are a group of cells called the quiescent center, which signal to the true meristem cells immediately surrounding them, which then, which retain this undifferentiated condition. And that undifferentiated condition is maintained by signals from this group of cells here called the quiescent center. But then those stem cells will divide to form all the other cells of the root, the vascular tissues up the middle, the epidermis up the side, and the root cap down below. Now, unlike the shoot apical meristem, which produces lateral organs off the side, the leaves, for example, the root apical meristem doesn't do that. But there are lateral structures produced in roots, not from the meristem, but from previously differentiated cells, and these are the lateral root meristems. So this is a sort of a form of redifferentiation. This is an example of the plasticity in development that plants exhibit so beautifully in a way that animal cells don't really do. The ability for differentiated cells to reorganize and redifferentiate into other cells, including stem cells. So this is a new meristem that's forming off the side to form lateral root branches. And it's perfectly nicely organized, even though it's formed from these previously differentiated cells. So there's a whole reprogramming of differentiated cells to form new stem cells, and then ultimately more differentiated cells as that root develops. And then the third kind of meristem I want to just mention very briefly is called the vascular cambium. So this is a cylinder of cells down the longitudinal axis of the plant. And these cells, again, they all look pretty much the same. undifferentiated, but they will divide, and off the flanks they will expand and differentiate into the xylem and the phloem and cause that radial expansion of the stem, and gives rise to the woody tissues that we see in trees. Now, I've kind of indicated this idea of plasticity in development that is very clear in plants, but much less clear in animals. And any gardener will be able to tell you that you can quite easily visualize this kind of plasticity in development and see evidence of it just by taking cuttings. So this is a begonia leaf drawing of, and when you chop it off the plant, it's able to regenerate completely new root systems from previously differentiated leaf tissue, in this case the petiole down here. And plants have this ability to generate new structures from previously differentiated structures. So these are adventitious roots, induced at a site of wounding in this particular, on this particular stem of this particular plant. Here are other examples of adventitious roots formed in the wrong place, if you like. They're not produced underground, but these are produced off the stems of this particular tree. And experimentally, you can investigate this plasticity by tissue culture experiments, where you can take a bit of leaf tissue and you can culture it. And when you first put it into culture, it forms this wound tissue, which is called a callus tissue. So just cells will proliferate as this callus. But if you treat them in the right way with the right sort of hormones in the medium, you can get them to re-differentiate into completely new structures. They'll form new meristems, either shoot meristems or root meristems. And this is a technique that's really quite valuable for understanding both the molecular mechanisms that underpin the formation of meristems, but also has practical applications, which I'll talk about a little bit later on. But one example of how it's been useful is in this bit of work that was published a few years ago now, where some archaeologists were digging through the permafrost in Siberia, and they found some sort of frozen fruit that turns out to be 30,000 years old from this species Silene stenophylla. And although the fruit wasn't able to produce viable seed, they were able to use tissue culture techniques to produce firstly that callus tissue, that wound tissue, and then regenerate from that perfectly viable plants. So these Silene plants here are sort of clonal copies of the original 32,000-year-old Silene plant that was buried in the permafrost. So you can see that this kind of technique is quite useful in conservation work if you need to rescue species that are otherwise sort of becoming extinct. If you can retain these tissues for a long time, maybe frozen, and ultimately regenerate plants from them. And this happens through the formation of new meristems. So the meristems, again, are pretty important to the strategies that plants have for developing. And meristems can change their identity. So another example of the plasticity that we see in plant development. And the example I'm going to talk about in relation to identity change is in the shoot apical meristem. So as I showed before in a slide, this shoot apical meristem divides slowly, but it will divide. There's more division at the flanks, and at the flanks of this meristem are new leaves formed. And this is a scanning electron micrograph of the top of a shoot apical meristem, and then the new leaves are formed in a kind of spiral, in some species anyway, this spiral arrangement. It's quite a neat arrangement. And so that's the first leaf there, the second leaf there, the third leaf there, the fourth leaf there. And what's kind of interesting, and this has occupied the minds of mathematicians for quite a long time, is that the angle between adjacent emerging leaves, so the first one and then the second one, and the third one, is always 132.7 degrees, that angle. So there's sort of interest in why that might be the case. It's known as the golden angle. Anyway, that's a slight aside, but this meristem here is producing leaves, but under certain conditions, the meristem can completely change its identity, and instead of producing leaves off the flanks, it will produce floral meristems off the flanks. And in that floral meristem, the meristem will produce floral organs, so sepals, petals, stamens, and carpels, which are characteristic of a mature flower. So the meristem identity is changing in the sense that it is producing different structures off the side. And this is an example of an Antirrhinum snapdragon, where you can see that you've got the youngest meristem at the top. It's producing these floral meristems, and then these floral meristems are producing the different floral organs in the mature flowers. So, you know, the questions that bother people in biology at the moment is in plant biology is how does this work? So you've got very controlled rates of cell division and patterning of structures from those dividing cells. And this is regulated by, you know, genes being switched on and making proteins. And the activation of those genes in the right place at the right time in a spatially and temporally controlled way is mediated by internal hormone signaling systems. And these in plants are primarily determined by environmental triggers. So again, one of the kind of important sort of concepts in thinking about plant developmental biology is the relationship between the environment, the wider environment, and how that affects hormone signaling systems, how that then activates genes and then proteins to ultimately give rise to these developmental consequences that we see. So I'm going to just say a little bit about the environment, because this is really the key to understanding plants. And in terms of flowering, for example, two major environmental factors for many species, not all species, are the length of day and temperature. And these have evolved as mechanisms to allow the plant, in this case this is an Arabidopsis plant, our favorite model organism, to determine the time of year. So this plant here would typically flower in the spring. And so during the short days, you know, so this would be an 8-hour day and 16-hour night, that would be typical of the winter time. But in longer days, which could be either in the spring or the autumn, the thing will flower. But it doesn't flower in the autumn because another environmental factor that's a level of regulation that's layered on the day length regulation is temperature. So if the plant has been exposed to cold for a long period of time, it will flower. In other words, when it's been through winter, it will flower in the spring. But if it's had a summer of warmth before that period, it won't flower. And that's how it differentiates between long days and short days, or the long days that could be either in the autumn or the spring, because of the temperature of the preceding few months. And the molecular mechanisms underpinning that are quite well understood. So some species are blind to day length, especially tropical plants, ones that are around the equator where there is really no big difference in day length. So things like rice or maize, tomatoes, which originated in Central and South America, these are blind to day length. But a lot of the plants that naturally occur in northern and southern hemispheres, they're regulated by this combination of day length and temperature. Other sort of examples of environmental triggers are things like mechanical stress. So this plant here, this tree, has developed a peculiar shape because the wind is coming from the left-hand side, and it's not that it's sort of blown the plant so that it's just, you know, wobbling about at an angle. It's actually a developmental response that's seen in the way in which the cambium, that vascular cambium, works. So you've got greater division and expansion of wood formation on one side and less on the other, which is associated with the mechanical force of wind over a period of years. So the environment's really important. And what I'm now going to do is talk a little bit about our favorite meristem, which is the root meristem. We're interested in roots. This isn't the plant we work on, but I took a photo of it in China because it's got a nice set of what are called buttress roots. These are things that hold up the tree to stop it falling over. But this plant's kind of interesting because it's considered probably the most toxic plant out there. So it grows in China and Southeast Asia. And in China, it's known as the 7 up, 8 down, 9 death plant, meaning that if you are poisoned by chemicals that are found in the bark of this plant, and some tribes in Southeast Asia use this as a sort of an arrow poison. It means that you can't take more than 7 steps uphill, 8 steps downhill, or 9 steps on the level before you drop dead. So that's anti-iris, and it produces chemicals called cardinalides, which basically stop your heart working. So yes, we're interested in roots. Some other, so just a plant that's fallen over, but I like the root system. Why are we interested in roots? Well, they're biologically, of course, they're very important to plants. They allow plants to exploit the environment. These are roots of a mangrove, a tree in a mangrove swamp, which is a highly saline environment. And these roots have evolved to exclude what would otherwise be toxic concentrations of sodium chloride from this mangrove, and the plant goes perfectly happily. So salt exclusion is one thing that roots do. Of course, they also try and grow down to find water and nutrients. They keep the plants, like those buttress roots, they keep the plant upright as well. So biologically, they're really important. And the architecture of the root system, by which I mean sort of the number and length of the main roots and the branched roots, is under a lot of environmental control. So the architecture of the root system will vary according to whether the plant's under drought conditions or not, whether there's enough nutrients in the soil or not, whether the soil is compact or loose or not. And roots behave quite interestingly when they encounter a mechanical barrier in the soil, maybe a rock or something. So this is a video that was taken by one of my PhD students who shows an Arabidopsis root growing down and hitting a barrier. And what's happening is that the cells in the meristem, which are down here, again, will keep dividing, so the root divides, but then the cells further back expand, and they expand differentially to give this sort of characteristic shape as the root's trying to grow down towards gravity, but trying to find a way through. So the way in which roots respond is influenced by these kind of mechanical forces as well. So as well as roots being important for exploiting the environment, they are also themselves exploited. in this case by parasitic nematodes, which invade roots of a number of species, and they work their way usually between the cells, actually, to cause minimum damage, so the plant can't activate a kind of immune response. And then they feed on the vascular tissues that are in the middle, and they actually induce a redifferentiation of those differentiated vascular cells to form what's known as a feeding structure. And you can see the shape of those cells has changed because these nematodes actually release chemicals that act a bit hormonal-like, and they mimic plant hormones to cause the redifferentiation of those cells. And of course, this is important because that root damage can cause crop losses, so it's an important agronomic trait. Anyway, we're interested in the way in which the meristem is both constructed and functions. And what's interesting in this root, this is an Arabidopsis root, is that you've got this sort of linear arrangement of processes that normally occur across time. So areas of cell division followed by areas of expansion followed by areas of cell differentiation. So that's the sort of progression of the formation of differentiated cells from those stem cells down here. And the meristem is the key site of cell division, but the question is how does it work? Now, this is an Arabidopsis root meristem, root tip, but not all roots have looked like that through the course of history. So this modern root tip, of which Arabidopsis is one example, is quite different from ancient root tips. So the thing about Arabidopsis is it's got this root cap, and this root cap is important as a gravity sensing structure. I can talk about that at some other point. But if you look through the fossil record, this plant here called Astraxylon, which was the fossil was discovered in the Rhynie Chert, which is a sort of geologically very old kind of fossil bed, 407 million years old, has no root cap. So the oldest plants, and this is the oldest root meristem found to date, has no root cap. Whereas 100 million years on, this is a 300 million year old root, you can see that it's evolved the root cap. So not all plants always had root caps. And in fact, this root cap, which is representative of the modern root, probably arose independently through evolution. So there was probably a rootless common ancestor of the so-called vascular plants, the bigger plants that are around today. And there's one lineage that Astraxylon represents a member of, which had no root cap, but then it's given rise to plants that do have root caps, and then independently other species, modern species, also have a root cap. So the development of the root cap is an example of convergent evolution. Obviously has a strong selective advantage, probably in terms of gravity sensing, and also maybe some mechanical protection of the delicate stem cells, which would be in that yellow region there, to protect that. Those ancient plants often had roots that, I mean, even older than Astraxylon, they had roots that, well, weren't proper roots. They were kind of hair-like structures. So things like this here, you can see they're more like the hairs that you see on a root. If I go back a couple of slides, if I can do it. These are root hairs, and they're there to sort of increase the surface area of the root. And ancient plants most likely, well, you can see them here in the fossil record, they're nicely preserved. The roots there, which are known as rhizoids, acted like roots. They were water and nutrient absorbing structures, but they weren't proper organized roots as you and I would know them. And this is another example here. And modern plants like mosses and liverworts also have retained that primitive system, this kind of rhizoid system. So this is some examples of modern mosses and liverworts. Mosses here, again, you've got these sort of little rhizoid-like structures here, hair-like structures. But most so-called higher plants have properly developed roots, and there are a range of structures. From this is an Arabidopsis root system in the soil. We will recognise a carrot. That's a root that produces little roots off the side. And then the architecture of cereals like rice and maize is broadly similar, but subtly different in its organisation. And so the question really, both from an evolutionary point of view and also from a sort of a mechanistic point of view, is how is the meristem regulated? How are the cells patterned and the activity of that meristem maintained? And that's really the focus of the work in our lab. We use this Arabidopsis root system, and what you can see in this sort of cutaway 3D model of the root tip is how neat it is, the organization of cells. And the organization of these cells is pretty stereotyped in Arabidopsis. Now, when we started off this talk, I talked about this plasticity, this variability in plant phenotype. You know, you can't predict the number of roots or the number of leaves in a plant, but what you can predict is the patterning of the cells in the root meristem, for example. And what you see here is those quiescent center cells that play a critical role in controlling the stem-like behavior of the cells immediately surrounding. So these cells here, this green cell, these cells immediately below are the true stem cells. And how are these controlled? How is that pattern controlled? And how is the division of those cells regulated? Because it is regulated because these plants are able to grow in quite predictable ways in response to particular environmental signals. So what we're doing in the lab is trying to identify the genes that are important in maintaining that pattern of cells, the activity of those cells, and also the hormonal signaling networks that activate those genes. And that's really the focus of our work. So one hormone, which I guess some of you will know about, is probably the most well-known plant hormone. Auxin, indole acetic acid, is the sort of most common form. And one of the things about auxin is that it moves from cell to cell in a highly regulated way, controlled by proteins that control its influx into the cell and efflux out of the cell. And what that means is that you can set up gradients of auxin concentration. So auxin is actually produced in the developing leaves primarily, I mean in large concentrations, produced in the leaves and shunted down the plant to the root tip and to other parts of the plant. And it moves around, auxin moves around by virtue of those transporter proteins to form this sort of inverse fountain where it accumulates at the highest concentration around the center of the meristem. This is where the stem cells are. So now that's not just a coincidence. This concentration, this high concentration of auxin here, is responsible for the maintenance of the stem cell identity and activity. And that happens because auxin activates two, well, in fact, more than two, but an example here is two transcription factors. These are genes encoding proteins that regulate cascades of other genes. They're called the plethora genes, and this represents the localization of their messenger RNA in the root tip. So plethora 1 is in this group of cells here, around the center of the root meristem. This is plethora 2, has a slightly different localization of its RNA, but there's an overlap here. And we know that these two genes are important. We know they're activated by Auxin, but we know they're functionally important because if you generate mutants in these genes, so you screw up the development of the root. So this is a single mutant of one of the genes, plethora 1 gene, you don't see much of an effect. Plethora 2 gene, you don't see much of an effect. But when you knock out both together, you see a dramatic effect on the development of the root. And you don't need to be an expert on root development to see that this root here looks pretty sick compared with this normal looking root here. So what Auxin is doing is activating genes, key genes, master regulatory genes that activate a whole lot of other genes that are required for the maintenance of the identity of the stem cells. And in this double mutant, you've lost the stem cell activity. So the root dies, and that's what you end up with. So we're interested in how genes and signaling networks work together. And I'm just going to sort of very briefly mention one of our favorite genes that we've been working on, and the hormones, the hormone networks that we've been working on. So I mentioned Auxin as a key hormone. It accumulates around this part of the root meristem. There are other hormones that also affect the development of the root, whether it's maintaining or disrupting the identity of the stem cells or just regulating the division. So one is Auxin, another is called cytokinin, another is ethylene. And these hormones also interact with each other. So Auxin can induce ethylene biosynthesis. Ethylene biosynthesis can regulate Auxin synthesis, ditto cytokinin, and it can be regulated by ethylene or regulate ethylene. So you've got a very complex network, so hence the word network here. You've got a network of these interacting hormones that if that network is perturbed and you get too much ethylene, ethylene will block the growth of the root. So you need a careful balance of these activities to stop too much ethylene accumulating. Ethylene is a gas, but it acts as a hormone. And too much of it will stop the roots growing. Now, we identified, and this is a common way of understanding the molecular mechanisms involved in plant development, looking for mutants. So we found a mutant, which we call the Polaris mutant, which has a very short root system compared with the normal wild-type plant. And we cloned the gene that was defective in this mutant, and it turns out to encode a short peptide, 36 amino acids peptide. So a very short protein. This is a protein sequence. Question is, how is that required for controlling the activity of the root? It controls the meristem activity. And it's taken us about 20 years to figure out what's going on, I have to say. But what this Polaris peptide is doing is actually blocking ethylene responses. So in fact, this short peptide works by being a regulator of the ethylene receptor protein. So ethylene is detected as a hormone because it interacts with a receptor and then activates a whole load of other genes. And what Polaris is doing is interacting with that ethylene receptor to block its activity. So when you lose that Polaris function, when you mutate it, you get too much ethylene and you get the short root that we saw in the mutant here. This has got too much ethylene signaling going on, so it's a short root. And the Polaris peptide is acting to limit that process, that inhibitory process. Now, just to make things more complicated, Polaris is itself regulated by Auxin and by ethylene, and indeed by cytokinin. So if you're trying to understand the balance of activity across this network, your brain starts to overheat. You can't do it. You can't think about how these interactions work. So this is where the mathematical modeling comes in. So what we've been doing, and when I say we, I don't mean me at all, I mean Junli Liu in our department, who's a very good systems biologist, has helped us try to unravel the regulatory network that is occurring in the root tip. So the way we do this, and again, when I say we, I mean him, is you first of all sort of set up this kind of network, and you put in the interactions that you know about. So auxin regulating cytokinin, cytokinin regulating auxin, auxin regulating ethylene, Polaris is in there, ethylene signaling's in there, etc., etc. So there's a sort of simple network, but it doesn't really tell you how it's working, and it's not testable as such. So you have to construct a mathematical model. And this is where Junli comes into his own, because he's able to develop kind of mathematical representations of the rates at which auxin moves into cells, the rates at which it's made in cells, removed from cells, the rates of transcription of the Polaris gene, conversion of receptors into one form or another, etc. And when you put all that together, you come up with something that is represented by this sort of network of interactions, this mathematical model, which you can run on a computer, and you can test the interactions between these components. So, for example, you can say, right, what happens if you change the amount of auxin in this system? How does that affect cytokinin levels, ethylene levels, Polaris levels, and so on? Or you change cytokinin, how does that affect the other components in the network? So this is, so when I first started talking to Junli about this, I was a bit skeptical that by introducing a relatively small number of components into the model, we would actually be able to get anything that was really representative of what was going on in a real plant. But I was wrong, because these models are incredibly powerful. So this was our first attempt to, with one of my PhD students, Simon Moore, develop a kind of a mathematical simulation of what's going on in a root. And this was the first attempt. This was a sort of a diagrammatic representation of a root, with the root cap down here, the sort of epidermis, cells here, vascular tissues here. And within each of these cells was embedded those parameters that were described on the previous slide, you know, the auxin going into cells, the rate of auxin synthesis, and so on. And that model that I had in this slide here was embedded in each cell. So that model was embedded in each cell, and there's 80,000 data points in this model here. And you can use that to try to predict how a root is going to behave. And it worked pretty well. But of course, this doesn't really look very much like a real root. So we took it to the next level, where this is actually a much more realistic looking root. And that model was embedded in each of the cells in this, what's actually a confocal image of a root. And what the model has done is made a prediction of the distribution of auxin in the root tip, based purely on those interactions in the model. And this is a heat map of where auxin is accumulating, and you can see that it's at the highest level in that stem cell niche. So that's a pretty good representation of what's actually happening in a root. And if you compare the predicted auxin levels in different cell types in the root, and you compare it with what's measured in those cell types through experimentation, you find a very good correlation. So this is quite a complex model. It's got 375,000 data points in it, but it's starting to simulate very nicely aspects of the root. In this case, the distribution of auxin, but we've also looked at the distribution of other hormones. And then in this slide here, what we've done is simulate the expression of a gene. This is that Polaris gene. This is its expression as a fusion with the GFP, the green fluorescent protein. So you can visualize where the protein's localized in the cell. And this is the model that was a simulation based purely on running that simulated model. And you can see that I think it ties up pretty well with what you actually see experimentally. So that's the kind of experimental approach we're using. We're using sort of mutant analysis, functions of knockouts mutants combined with this mathematical modeling, which allows you to think up new experiments based on the model not doing quite what you think. So you can test the model and come up with whole new kind of experimental approaches. So it's working really well. So I'm just going to finish off by sort of stepping back a little bit and asking, why have meristems evolved in plants? Why have plants adopted this very different approach to controlling their growth and development compared with animals? And this guy here, Theodosius Dobzhansky, is a famous geneticist who, in an essay, made the point that nothing in biology makes sense except in the light of evolution. And so when we're thinking about why plants have meristems and animals don't, we need to think about this in the light of evolution and what selective advantage of meristems is. And well, one selective advantage of being an animal is that you can run away from stressful conditions, but plants can't do that. So if a plant is set upon by a herbivore, like this little bunny rabbit or this rather angry looking sheep, it has to evolve a different kind of approach to dealing with that mechanical damage that's going to be inflicted by that sheep. So what plants have evolved is a system of, if you like, emergency meristems, these so-called axillary meristems that are sort of further down the stem. And when this top bit of the stem gets chewed off by an angry sheep, these new meristems are then activated and the plant survives. So plants have evolved these developmental approaches to cope with predation rather than behavior, which has been evolved by animals to avoid these kind of stressful situations. Plants have this inherent plasticity, this flexibility in their development, which allows them to survive in adverse environmental conditions. So we see this activation of dormant meristems, or we get the activation of new meristems following wounding or other stresses. And finally, why does any of this matter? Well, there are some important questions in biology that are addressed by looking at the way in which plants have adopted this kind of very different approach to growth. The questions are, how have these different growth strategies evolved? How do the genes control the identity and function of these tissues? And what is the link between genotype and phenotype? And these are sort of big questions in biology, whether you're a plant biologist or an animal biologist. And we're starting to be able to address these by different strategies. There are also practical values of understanding meristem function. So the science underpinning meristems is really the basis of the micropropagation industry. So if you go to Marks and Spencer's and buy an orchid, it will have been generated not from seed, but by micropropagation. So micropropagation is where you can take a meristem, as I showed in one of those earlier slides of roots in culture and shoots in culture. You can take a meristem and you can propagate multiple plantlets from that single meristem. So you can get rapid propagation from a limited amount of starting tissue. And like that example of the plant that was rescued from the Siberian permafrost, it provides a way of conservation for rare species. And also this sort of plasticity of plants and the ability to just generate new meristems is the basis of plant genetic engineering. So you can take these color cells and you can introduce a gene into one of them and you can regenerate a whole plant. And if that gene is integrated into the chromosomes, the entire plant will contain that new transgene. And that's the basis of plant genetic engineering. Relatively straightforward these days for many species. The other sort of thing is meristems determine the architecture of plants, which is of agronomic importance. So farmers, you know, they develop machinery to harvest plants that have particular architectures. Maybe you can improve yield by regulating shoot branching. Maybe you can improve yield by regulating the architecture of the root systems in response to a lack of certain nutrients or lack of water. So the underpinning science is actually of some practical value. Where's all this going in the future? We're rapidly developing new technologies, new molecular tools to allow us to understand the genes that underpin important traits that affect the development, the architecture of plants. Mathematical modeling can help us understand how genes and hormones interact together to understand mechanisms better. If we understand regulation at the molecular level, that can help breeders screen for genes that are associated with good traits, architectural traits, altered branching, flowering, root traits, meristem genes, you could call them. So breeders can benefit directly from an understanding of that basic molecular biology. And we can also identify genes associated with other aspects of development like cell expansion and differentiation, things like, so I should say, cotton fiber quality, oil yield, fruit ripening, and nutrition. So that's pretty much all I want to say. Just would acknowledge a few of the people that have done the work. This is Junli, here he is. Junli did the modeling work. Simon did the modeling work. Vinny, as he calls himself, has done a lot of the modeling work. And the others are all doing different aspects of root development work.